Good morning, all of you. Today's class is on the basic embryology. So, we will be talking about the development of the fetus, its various stages. Before that, we will talk about the male and the female gametes, how they fuse, and how the implantation of the fetus takes place, then how it develops. So, let's go into the basics. What is embryology? So, it's a branch of anatomy which deals with human conception, intrauterine growth, and development. So, it's also a study that will let us know about the congenital anomalies and the development of the various anatomical structures. So, whenever there is a variation in the normal development, we use the word anomaly. Anomaly means some abnormality in the normal growth. So let's go into the basic terminologies first. The word zygote means fetus. So how the zygote is formed? It's formed by the fusion of the male and the female gametes. So the male and the female gametes are the two pronucleuses with haploid chromosomes, say 23X chromosomes. What is the total number of chromosomes in human body? It's 46, right? So how is that 46 derived? 23 plus 23. So when a baby is formed in the womb, it is formed by the fusion of the male and the female gametes. So the female gametes produces the ovum, that is from the ovaries, and the process by which the ovum is formed is called ovulation. The ovulation occurs from puberty to menopause. So in female, the ovulation is continuous and it occurs from puberty to menopause. How this ovum is actually produced? The ovary has number of primary ovum or primary follicles that are arrested in the first meiotic stage division in the diplotin stage. So what is this diplotin stage? The meiotic divisions have meiosis is a process of cell division. This word itself may be new to you. What is meiosis? There are two types of cell division that takes place in the body. One is the mitotic division and the other is the meiotic division. So in the first meiotic division itself, the own primary follicular division ceases at the diplotin stage. So all the primary follicles in the ovary are in the stage of the primary follicular stage. It is not progressing to the secondary follicular stage because at that stage itself it is arrested when the child is in the embryonic stage or in the fetus. So the ovum consists of number of primary follicles present. Okay? So the primary follicle only later matures into a secondary follicle and it develops into a ovum. Okay, so the terminologies that may be new to you here will be the mitosis, the meiosis, the primary follicle, the secondary follicle, and the diplotin is one of the phases in the meiosis. Okay, so at this stage the primary follicle development is arrested, and at the diplotin stage itself they remain in the ovary. When the fetus completely develops, when the child is grown, and when it reaches the puberty stage, primary follicle start to mature into secondary follicle and they start to release the ovum. So this happens in puberty. Now, what are the important organs that we will talk about today in the female reproductive system? One is the ovary, uterine tubes, the uterus, the cervix, vagina, and the ovum which is produced in the ovary is carried to the uterus by the uterine tubes. First, let's talk about the ovary. The ovary is a female gonad that is responsible for the production of the ovum. So once the ovum is produced in the ovary, it is transported through a tube called as the uterine tubes, which bring the ovum to a hollow organ called uterus. This uterus is a pear-shaped organ, hollow muscular organ, which has a thick muscular wall. And that thick muscular wall is thrown into three different layers. One is the the perimetrium, the myometrium, and the endometrium. So here, the thickened muscular wall also plays a favorable location for the zygote to develop. So the uterine tubes are the muscular tubes that bring the ovum from the ovary to the uterus. The word fertilization is the term that explains the fusion of the male and the female gametes. That is the ovum with the sperm. The ovum that was released from the ovary travels all along the fallopian tube, and when it reaches a part of the fallopian tube called ampulla, the fertilization can occur. Then, after the fertilization, it is no more called ovum, it is called a zygote. So later, they undergo development, two cell, they undergo cell division to form two cell, four cell, 8 and so on, and the cell division actually takes place. It turns into a 16 cell stage called morula. Then, after that, it reaches the uterus, and in the uterus, it undergoes implantation. So, this is the phase. This is just a brief of the phases what is going to happen during the fertilization and implantation. So, the uterine tubes are nothing but the muscular tubes that bring in the ovum from the ovary to the uterus. The uterus is the hollow muscular organ which is ready to receive the fertilized ovum for its growth. It's a quite big organ with a thick muscular wall. Okay. The lower end of the uterus is called as the cervix, and the cervix continues lower down as the vagina. So this becomes a passage for the sperm to enter into the uterine tube. The uterus. So the vagina is the outermost uh, opening of the female genital tract, and through the vagina the passage continues as the cervix. The cervix is the lower end of the uterus, and through the lumen the sperm enters into the tract. So the ovum that is produced in the ovary is carried to the uterus by the uterine tubes, and the lateral end of the fallopian tubes open itself into the peritoneal cavity, which means the fallopian tube here it makes the female genital system or the female peritoneal, it is open to the female peritoneal sac, hence we call the female peritoneum as open sac, because it is open to the exterior and it is one side it is exposed to the peritoneum directly through the fallopian tubes. So that we call it as an open type of peritoneal cavity in female, while in male this is closed. So now how the sperm enters into the female system. The ovum that is produced in the ovary enters the fallopian tube and reaches the uterus. The sperm enters the uterus through the vagina and cervix. Then it fuses with the ovum at the uterine tube. So this image clearly shows you the uterus. This is the hollow muscular tube of the uterus, and this is the fallopian tube that is going to bring in the ovum. This is the ovary. So if you find the ovum needs to enter from the ovary through these ends called as the fimbriae. The fimbriae are usually not smooth. They are finger-like projections which connect to the ovary, and they help in bringing the ovum from the ovary. Then the next broader portion of the fallopian tube, which is stained pink here, is called infundibulum. So this is a broad area to receive the ovum. Then the ovum reaches this greenish phase here, what we call it as the ampulla. This is a place which is very favorite for the fertilization, and once it crosses the ampullary stage, if fertilization occurs, then it is no more called ampulla. No more called zygote. No more called ovum, but it is called zygote. So the zygote undergoes cell divisions. First cell division, becoming 2 cell stage, 4 cell stage, 6 cell stage, 4, 8 cell stage, 16 cell stage, so on, and it becomes a blastocyst. So this blastocyst moves through the isthmus and enters the uterus. So around 5 to 7 days after fertilization, that is, the fertilization has occurred in the ampulla. After fertilization, the zygote travels through the passage of fallopian tube, and 5 to 7 days after fertilization, it's implanted in the uterus. So this is the diagram of the entire system, which clearly tells you the phases of ovulation. So this is the ovary that has been shown as cross section, which has a number of primary follicles. So you see number of rudimentary primary follicles in various phases. So these primary follicles, which has been arrested in the diplotin stage, one of them, among the many follicles, primary follicles that are seen in the ovary, one of them starts to undergo maturation. So once it matures, it comes to a stage where it is covered by an outer cell layer, an inner cell which has the ovum, mature ovum. And here there is also a cavity which is seen outside the ovum, so that is filled with the fluid. So once on rupture, the ovum is released. And once the ovum is released, it is called corpus luteum. The corpus luteum is usually yellowish in color. It's called carotene stage. And this corpus luteum is usually yellow in color because of the pigment which we call it as which is yellow in color, which gives it that pigment. Okay. Okay. From there, if the ovum that is released is not fertilized, it is going to disintegrate. If it is fertilized, then what is going to happen? See, the ovum that has been formed is released into the fimbriae part of the fallopian tube. It enters into the ampulla, then it reaches the isthmus, and here when it reaches the ampulla, then the ovum will meet the sperm. So if it meets the sperm, it is called as a zygote. It is called as a zygote. And the zygote undergoes cell division, starts its cell division. The first cell division is the 2-cell stage, then it becomes 4 cell. After 4 cell is 8 is becoming 16, so on. And once it reaches the 16 cell stage, it is called as the morula or the mulberry stage because it looks like the fruit, mulberry fruit, with number of cotyledon-like appearance on its exterior. It moves through the fallopian tube and enters into the uterus. Here it undergoes implantation. So at the implantation, see, this is the site where the implantation occurs. So once the implantation is occurring, then the development of the zygote takes place. Before the zygote reaches the uterus, the endometrium that lines the uterus needs to undergo number of changes. Endometrium is the inner lining of the uterus. Inner lining means the mucosal lining of the cavity of the uterus. So the endometrium usually is very richly vasculated, and that undergoes a number of changes, morphological changes, what we call it as the decidua. So decidua actually will increase the vascularization in that particular area. It prepares the place, the endometrium, for receiving the zygote, so that the zygote will receive nutrition. Zygote will become well nourished through the vasculature from the endometrium. So this is the structure of the uterus, and this is the endometrium, this is the myometrium, this is the area called as the cervix, the lower end of the uterus, this is the cut section of the vagina, and this is the luminal wall of the uterus. So if you clearly see here, there is a covering of the a connective tissue covering around the ovary, right? So that connective tissue covering, we call it as the mesovarium, and there is also a tissue that covers and attaches the fallopian tube, so that we call it as the mesosalpinx. The uterus on its lateral wall has a sheet of tissue that attaches it to the lateral pelvic wall, and that we call it as a broad ligament. So these ligaments give support to the uterus so that it is able to hold the fetus. So first, let's talk about the ovarian cycle first. How the ovum is formed and how it is taken from the ovary to the ampulla of the fallopian tube for fertilization. Carefully listen. First, the maturation of the female germ cells happens, okay, and the stromal cells of the ovary will provide a protective outer coat outside the female germ cells. Now, the female germ cells are nothing but the primary follicular cells present within the ovary. When are these follicular cells formed? It is formed when the female child is in the uterus itself, in the mother's womb. So in the mother's womb itself, the primary follicles are formed, and they are arrested in a diplostene stage, and at that particular stage it is waiting. Only at the puberty, the follicular stage, the primary follicle starts to mature first time, and that we call it as the puberty, or what we call it as the maturation. So then from that stage up till menopause, the female will frequently have the menstrual cycles, and the menstrual cycle will mark the period which she is fertile to bear a child. So fine. Now the ovary. Now let's talk about the female germ cells, which is seen in the ovary. So that has an outer covering called as a stromal cells that provides the protective outer coat. The primary oocyte is a single layer of follicular cell, what we call it as the primary ovarian follicle. So the primary germ cell can also be called as primary oocyte, and the follicle that covers it, follicle means its outer structure, for the cells that cover the primary germ cell is called as the primary follicular cells, and we can also call it primary ovarian follicle. So this further develops and it is going to undergo the second stage. The second stage is called as the secondary ovarian follicle. This primary ovarian follicle is getting converted into secondary ovarian follicle. Later, a cavity tends to appear within the secondary ovarian follicle, and that cavity is also dividing the cells into two layers. That is one outer layer and the inner layer. So the cavity appears like it has an outer follicular cells, and at this stage it is called the tertiary follicle. The secondary follicle now has undergone cell divisions and has reached the third stage, what we call it as a tertiary follicular stage. Now, from this tertiary follicular stage, the matured follicle, that is the matured tertiary follicle, will release the ovum, and then after that, the matured follicle will turn in, and once it reaches the stage of releasing, it is called as the Graphian follicle. So here what you find is the image of that Graphian follicle that is fully matured to release the ovum. So the outer layer is called as the theca externa, the inner layer we called as the theca interna, and every layer of cell has its own functions in secretion. So the main primary function of these cells in the ovum is, one thing is secretion of some enzymes. Two, it is going to nourish the ovum for the nourish the ovum for its development. Third, it's going to be protective so that the ovum is not injured by any external factors. So these are the three functions of these cell lining layers in the Graphian follicle. Now first we will take the outermost layer, the theca externa. The theca externa is the outermost layer, and it is going to be called as the future trophoblast. And that is the layer which is going to further develop into the placenta, and that is the layer which will so this is the theca externa, and this layer is called as the theca interna. So this layer we call it as the connective tissue in between the theca externa and theca interna. So this is the layer which contains the inner egg cell in mature egg cell. So usually the estrogen is the hormone that is predominantly produced by the theca interna for its development. Theca interna mainly helps in producing estrogen that helps in the development of the egg to its fullest. And it also has a big antrum called as the follicular antrum, which is filled with the fluid to nourish the ovum. So next we are going to talk about another cycle called as the menstrual cycle. So this ovum is ready and it is released. The process by which the ovum is released is called as what? Ovulation. So once the ovum is released, it is going to get ready for fusion with the sperm. It keeps traveling from the infundibulum. It goes to the through the infundibulum. It reaches the ampulla. It is waiting for the sperm to reach it. If the sperm reaches, it is going to get fertilized. If it is not going to reach, then automatically the ovum will undergo degeneration, and it turns itself into uh, you know it turns into corpus luteum. Now What is menstrual cycle? So menstrual cycle is nothing but the cycle that brings in the fertile ovum ready for fertilization. So first, the menstrual cycle is basically 28 days, and the first 7 days, which is marked in red, is denoting the bleeding phase. Day 8 to day 12, we call it as the proliferative phase, where the ovum is growing. All the phases what we just now saw, isn't it? How the primary follicle is getting converted into secondary follicle, secondary into the tertiary follicle. So these stages are happening in the proliferative phase. They are happening in the proliferative phase. That is day 8 to day 12. Now day 13, after formation of the tertiary follicle, ovum is released. This ovulation is one of the important date we marked. That is from day 13 to day 15, the ovulation can occur. So it can happen between 13th to 14th day. The ovum gets released from the tertiary ovarian follicle. Now the ovum is released and it is fertile, waiting for the sperm to get fertilized. From day 15 till day 28, it is ready for fertilization. But the first 14 days will mark, and up till ovulation, if you see the ovum is released, it's released into the tract and it's waiting for the fertilization. If the fertilization does not occur, it turns into a stage called corpus luteum, and the corpus luteum is nothing but the disintegrated phase, and later it turns into a scar called corpus albicans. So once it is completely it is disintegrated, then it induces the next phase of cycle of next phase of bleeding, or the next cycle. So totally there are 28 days involved in one menstrual cycle. So let's talk about the features of the Graafian follicle, that is the tertiary follicle, which is having those two layers of cells and with a mature ovum. So the mature follicle differentiates into two layers, an outer theca externa and the theca interna cells. The theca externa are nothing but stromal cells that break, and the follicle later ruptures. So the theca externa has to rupture first. It has to rupture the stromal cells to release the ovum into the fallopian tube. That results in the shedding of the ovum, yes, the secondary oocyte. And this process by which the ovum is released is called as ovulation. Ovum matures, and the matured ovum is derived as an end product of the ovarian cycle. So once the ovum is mature, it is released into the fallopian tube. So this is again a diagram that shows you the primary follicle very clearly. This is the rudimentary stage of primary follicle. It is getting turned into the secondary follicle here, and the secondary follicle develops an antrum. Antrum means a cavity. So the cavity will differentiate it into an outer cell layer and an inner cell layer. The outer cell layer is called the theca externa and the inner cell layer is called theca interna. And this is the mature ovum. This antrum is filled with a fluid which is rich nutritive for the ovum. And once it reaches the matured stage, that is the tertiary follicular stage, the ovum ruptures and it is excreted out, or it is pushed out of the follicle. Once it is pushed out, the ovum is released into the fallopian tube, while the cell structure or the follicle that produced the ovum becomes corpus luteum, and this mature corpus luteum becomes the corpus albicans, and it becomes a scar and disintegrates. That's how the the follicle develops and releases the ovum, or otherwise called ovarian cycle. This is the diagram that clearly shows you the phases of ovum development, and it is called as the ovarian cycle. So in this class we have learned two cycles. One is the ovarian cycle, another is the menstrual cycle. Now let's talk about the oogenesis, how the ovum is formed and how it is receiving the chromosome. So the primary oocyte undergoes the first meiotic division, or the reduction of chromosome into half its number. Both the male and the female gonads are fertile pronucleases, and they usually contain 23 chromosomes only. They do not contain 46. Okay? So they are hence called what? Pronucleates. They are the cells that are going to produce a zygote or a fetus on fusion. Just because the two gonadal cells or the two sex cells fuse, they are able to produce the zygote. So the primary oocyte here is later developed into a secondary oocyte that has 22 X chromosome and one polar body. This division is unequal, so that the greater volume of the cytoplasm is passed into the secondary oocyte, and the secondary oocyte further undergoes second meiotic division, and second polar body is formed, and it releases and it becomes 22x plus once and second polar body. So here this secondary oocyte that has been formed is called as the ovum, and along with the ovum the second polar body is also received. Division occurs only if the fertilization occurs. The secondary oocyte that is there is called as the ovum, and it undergoes cell division only when fertilization occurs. Okay, so the secondary oocyte will undergo, for primary oocyte is turning into secondary oocyte by the first meiotic division. Thereby, it has 22 X chromosomes with one polar body. What is this polar body? So, in the unequal division of the primary oocyte, there is a structure that is formed with a lesser quantity or lesser proportion of cytoplasm on division, and which is not, I mean, this polar body is not fertile, and it is going to just pass on, okay, as one of the cell along with the primary oocyte, and the development of secondary oocyte in turn forms the secondary polar body. So it is not a fertile cell which is going to take part in fertilization. So what is this male organ? What what is the counterpart phase of reproductive cell development? So till now we were talking about the ovarian cycle, ovum development, menstrual cycle, how the menstruation occurs, how the ovum is developed in the ovary and it is released. Now on the counterpart, what happens on the male system? The testis is called as the male gonad, and it suspends in the scrotum by a cord called spermatic cord. The seminiferous tubules present within the testis will produce the spermatozoa. The epididymis is a coiled tube that is seen within the posterior aspect of the testis, and that is a place where the spermatozoa is being formed in the seminiferous tubules. So the testis, as it is like a sac, and it is suspended in the scrotum. How is it suspended? By means of a tube called spermatic cord. That spermatic cord is allowing the testis to be in the scrotum, and thus mainly the testis has some tubules coiled and in coiled present within that testis, what we call it as a seminiferous tubule. So the spermatozoa is actually produced in the seminiferous tubules, and within the seminiferous from the seminiferous tubules it is passed out. So from the single duct in the epididymis, it continues as the vas deferens. And the vas deferens is a muscular tube transmitting the spermatozoa from the epididymis to the prostatic urethra. Along with, as the spermatozoa that is formed in the testis is getting transported through the vas deferens, it is also being added with some secretions of the prostate. So that we call it as the prostatic secretions, as well as along with it you also have one more structure called seminal vesicle. The seminal vesicle also is a necessary gland that is going to produce the secretions of the semen, and the secretions of the prostate and the seminal vesicles are nutritive to the spermatozoa, as well as it is helpful in the maturing it. So thereby it forms the base or the thickness of the semen. The vas deferens, terminally, it will end into the ejaculatory duct. So the vas deferens is the tube that is going to carry this spermatozoa from the epididymis. As it keeps traveling through the epididymis, it receives the secretions of prostate and the seminal vesicles, which makes the semen thick and nutritive for the spermatozoa. And the vas deferens terminally ends itself into the ejaculatory duct. The male urethra becomes a common passage for the urine and the semen. So, this is the image that shows you the epididymis, the testis, and the seminiferous tubules. So the epididymis, if you clearly see, the testis is a rounded structure, and on cross section there is presence of the seminiferous tubules that are located coiled within the testis. So they are held together like a coiled tubes within the bulb of testis, and from there carries the epididymis, continues the epididymis. So the epididymis takes out the sperm that is formed, and it is carried through the vas deferens. So epididymis continues as the vas deferens, and vas deferens will carry the spermatozoa to the terminal ejaculatory duct. So before it reaches that area, it is also receiving another secretion from the prostate and the seminal vesicles. Later, along with the secretions of seminal vesicles and prostate, the semen along with the secretions and the spermatozoa that is formed from the epididymis reaches the ejaculatory duct, from which it is ejaculated through the urethral passage. So in male, the passage for the urine and the semen is common, and that is the common urethral passage. So this is the general image of in a general protocol by which the spermatozoa is formed. But if you clearly see what actually is happening, what is spermatogenesis? That is how the cells are formed, how the sperms are formed. Let's see the cell cycle behind it. So the primary germ cell, that was initially the primary spermatocyte. So here you can compare the ovarian cycle with the spermatogenesis. So there the oogenesis, how the ovum is formed, we studied. Here we will study the formation of the sperm. So this is the primordial germ cell. The primordial germ cell is undergoing the first meiotic division, where it divides itself into two primary spermatocytes. So primordial germ cell is another generation before the primary oocyte. So there we started the ovarian cycle with primary oocyte. You remember that? So those primary oocytes only were in the stage of diplotin stage, and they did not undergo this meiotic division. They were arrested in that stage, right? Later they developed into the secondary follicular stage. Secondary follicular stage to develop the ovum. But here, the even a generation before that, primordial germ cells are cells that produce these male fertile germ cells. So they undergo meiotic division to produce the primary spermatocytes, and that primary spermatocyte will undergo the first meiotic division. So the process by which the primordial germ cells give rise to the primary spermatocyte is mitosis. First division is mitosis here. Later the meiosis starts. So the primary spermatocyte undergoes first meiotic division to become the secondary spermatocyte. The secondary spermatocyte in turn undergoes second meiotic division to produce spermatids. The spermatids will continue as the developing itself into the mature sperm. So this is the structure of a sperm, where there is a presence of the head, the pronucleus, acrosome, mitochondria, and there is a middle piece and a tail. Basically, we divide the sperm structure into three: the head, the middle piece, and the tail. The head has a tip called acrosome, which contains hyaluronic acid, and it also has a nucleus present within the head. The nucleus is highly potent, and it has the presence of the uh, active phase of energy is needed. ATP, there is more concentration of ATP there, because active metabolic changes takes place in the head. And who produces that ATP energy? The mitochondria, which is seen at the neck level. So the mitochondria is located at the neck of the sperm, and it is the cell, energy producing agent for the cell. Then the middle piece continues lower down, and it ends itself as the tail. Coming to the structure of a spermatozoon, it consists of the head, neck, body, and tail. It measures 50 microns in its total length, and it may extend even up to 40 to 50 microns. The head consists of nucleus, which is covered by a cell membrane, and its upper part is enveloped by an acrosomal cap. It consists of a centriole with two cylinders. The centrioles are the place where the cell divisions. The centriole is the place or the site for cell division, isn't it? So the centrioles are located within the head, and the distal part of the centriole presents 9 filaments which continue to form axial filaments. So the head consists of the centrioles along with the nucleus, because the centrioles will always be located within the nucleus, and the centrioles will help in the division, okay, cell division. Behind the head, what you get will be the neck, which contains a mitochondria, which is responsible for the production of energy, responsible for the motility of the sperm. Sperm needs to move to enter into the female genital tract. So the energy production plays a very important role here. Unless the energy is produced, unless the motility happens, the sperm will not be able to travel through the female tract for fusion. So let's go into ovulation to implantation. So ovulation we have already seen. Now we will see how implantation occurs. Now we have also seen the male counterpart. So once there on the male counterpart, the sperm is ready. So on the female counterpart, the ovum has been released into the fallopian tube. So once it is released from the ovary into the fallopian tube, through the fimbriae of the fallopian tube, the ovum enters in the infundibulum, and from the infundibulum it is going into the ampulla. So ampulla is a place where the zygote meets the sperm. Once it meets the sperm, the process by which the male pronucleus, that is the sperm, and the female pronucleus, that is the ovum, fuses, is called as the fertilization. So once fertilization occurs, it is no more called ovum, instead it is called zygote. It is a single stage when the fertilization occurs. But once the pronucleus of the male fuses with the pronucleus of the female, it turns into a zygote. By the way, in the male, the semen consists of millions of sperm, and not all sperms are going to fertilize the ovum. Only one sperm succeeds in fertilizing the ovum. So though the semen has millions of sperm and it is entering into the female tract, only one sperm succeeds in fertilizing the ovum. The remaining is discarded, or once after fertilization, on the day of fertilization itself, the cells of the ovum will prevent multiple sperm entry, so that the fertilized ovum is not refertilized by another sperm. Okay? So then, after the cell, once it is zygote is formed after fusion, it is going into the two-celled stage. The two-celled stage exactly happens 30 hours after fertilization. Say after fusion, 30 hours after the fusion, it becomes two-cell stage. So until the first cell stage to two-cell stage, there are many sequence of enzymatic reactions that are taking place. So when we learn it, the science that deals with this is called as the developmental biology. So within embryology, there is another division, a branch of science that deals with exclusively the development of the fetus, that we call it as the developmental embryology or developmental biology. So developmental biology is fully concerned a number of enzymatic reactions taking place at this phase. So what happens is the cell wall, outer cell wall of the zygote prevents the further entry of the sperm. That is multiple sperm entry, and it closes the outer wall of the ovum tightly, so that further entry of the sperm is prevented. And the pronucleus, that is the sperm that entered the ovum to fuse with the nucleus of the ovum, becomes active and the fusion happens. So this fusion, for the fusion to happen and the first cell division to take place, it takes around 24 hours. That's why I said the first cell division takes place at 30th hour after fusion. So then the zygote is no more called one cell stage. Now it is going to become two cell now. Two cell stage. Later 2 is going to become 4. 4 is getting divided into 8. 8 cell stage. And the 8 cell staged zygote moves into the uterine cavity. And 8 cell stage becomes 16 cell stage, which we call it as a morula. The morula is again an important landmark there. Why? Because it is also called as the mulberry stage, and the morula happens, or the formation of morula exactly happens 3 days after fertilization. So 3 days after fertilization, by the time the ovum has reached, by the time the ovum, the zygote has reached the uterus almost. So it has travelled all across through the ampulla. It reached through the isthmus area, and here it has reached the uterus. So now it is 3 days after fertilization, and it becomes blastocyst. The next stage is called as a blastocyst. What is blastocyst? The blastocyst is the stage where there is a cavity formation within the morula, And that cavity, until morula stage, it is a solid cell mass. It looks like a ball. Okay. So in the blastocyst stage, what happens? There is a cavity formed within that morula, and that we call it as a blastocele. The blastocele differentiates into two layers. From there, the two cell layer differentiation takes place: an outer cell layer and inner cell layer. Same as what we learned in the womb development, right? Theca interna, theca externa. Same name is going to be here also. Theca externa, theca interna. So we call it as the outer, otherwise we can call it as the outer cell mass and inner cell mass. Both the terminologies can be used. So this cavity is called as a blastocele. The blastocele contains it contains the blastocele also contains a big coelom, what we call as a which is filled with liquid. And here the implanted blastocyst after 6 days after the it becomes the blastocyst, it is entering into the uterus and it gets implanted. So implantation exactly occurs 5 to 6 days after fertilization. What is fertilization? So again there is a definition. Fertilization is the fusion of the male and the female pronucleus in the gametes, and this occurs in the ampulla of the fallopian tube. Whatever I have explained now has been given as a definition. So it is a fusion of 23X plus 23Y in male, while for a female fetus it is 23X plus 23X. Again, both the male and the female pronucleus, if the fusion occurs between the X chromosome of the female and the Y chromosome of the male, the fetus turns to be a male fetus. If it is the X chromosome received from female and the X chromosome from the male, it becomes a female fetus. The male has X and Y chromosome, isn't it? So they can either contribute it through X or it can be a contribution from the Y. So the fertilized zygote moves with the cilia of the epithelium, cilia present within its epithelium. It moves to the uterus, and during the fertilization, 44X plus 44Y is restored. Because it is 23 plus 23, the zygote is now restored with 46 chromosomes. That is a normal chromosomal pattern of the human body. So that is restored. In a female, it is becoming 44X, or in male, it will become 44Y. Then the 2 cell stage further progresses to 4 cell stage, so on to 8 cell, 8 becoming 16, and then it goes into the 16th cell stage, what we call it as the morula. The morula is called as a mulberry stage, and it differentiates into an inner cell mass and an outer cell mass. The outer cell mass is called as the trophoblastic cells, and they become the future placenta. So at this stage, it is called as the blastocyst because it is having a big cavity there, and it is formed 5 days after the fertilization. The morula only changes into blastocyst. Okay, first stage is morula, that is the 16th cell stage. Then it becomes the blastocyst. So that is almost 5 days after fertilization. By the time the uterine endometrium is getting ready there. So till now the zygote has not reached the uterus. By the time the uterine endometrium is getting ready, by the changes that are happening in the endometrium to become into a decidua. No more it is called endometrium, it is called decidua because it is more vascularized and it is getting ready for the nourishment of the zygote. So the endometrium changes to decidua for receiving the zygote for its implantation, and it occurs 7 days after fertilization. So the stages of embryo development, we can divide it into 3 for the descriptive phase. First is the germinal period, second is the embryonical period, the third is the fetal period. So germinal period is what we were talking all about now. 1 to 3rd week of its development, we call it as the germinal period because there is no proper formation of the body, like there's only the basic formation of the structures happen up till this period, the germinal period. Embryonic period is a time when the full development of the fetus happens and the growth of it happens, 4th to 8th week. Okay, so a fetal period is a period where there is no more new development. So almost entire organogenesis is over from 4th to 8th week, and by the time all the organogenesis is over after 8th week to 9th month, it is just the increase in size. So that we call it as the fetal period. So the development of the fetus can be divided into germinal period, embryonic period, fetal period. The germinal period is all in the form, it is just in the form of cellular stage. 3 to 8 week period is in the embryonic stage. 9 weeks after up till birth, it is called as the fetal period. Let's talk about germinal period. The outer cell mass, which we call it as the trophoblast, differentiates and gives rise to an extra embryonic mesoderm. So I said the blastocyst stage itself, there is a formation of two cell layers, an outer cell layer and an inner cell layer. The outer cell layer we call it as a trophoblast. We call it as a trophoblast. So that trophoblast in turn undergoes division. In turn undergoes division. That is an outermost layer and an inner layer. So the outermost layer will become the extraembryonic mesoderm, which in future will develop into the fetal membranes, placenta, and the umbilical cord. While the inner cell mass, that was the I said the outer cell mass, which is outer cell layer, which is developing into trophoblast. The inner cell layer is getting developed into the embryoblast. Embryoblast is the cells that are going to form the baby or the fetus. So the outer cell mass is fully outer cell mass, which is called as the trophoblastic layer, undergoes division into a very much outer layer and the inner layer. The outer layer is called as the extraembryonic mesoderm, and second layer will be having the intraembryonic mesoderm. Okay, so the intraembryonic mesoderm will be concerned with the formation of certain structures, while the extraembryonic mesoderm is mainly responsible for the formation of placenta, fetal membranes, and umbilical cord. So coming to the germ layers. Now, till now, the embryo is in the blastocystic stage, and till now we only know two cell layers. That is the outer cell layer and inner cell layer. Outer cell layer is the trophoblastic layer. Inner cell layer is the embryoblast layer. Now, how this embryoblast is getting converted into a trilaminar germ disc? So first, now till now it is dilamina. Dilamina means just two layers. Now it's going to become three layer. Trilamina. What is trilamina? Tri means three. So the inner cell mass, which we called as the embryoblast, is getting developed into three layers of trilaminar disc, and the third layer, which is going to be inserted between the embryoblast and the trophoblast, is called as the mesodermal layer. Okay, what we called as a mesoderm. I told you the outer trophoblast gets divided into two, that is the extraembryonic mesoderm, intraembryonic mesoderm. So the extraembryonic mesoderm is concerned with the formation of placenta, umbilical cord, and the fetal membranes, while the intraembryonic mesoderm is concerned with the formation of the third layer, that is the mesodermal layer. So this becomes the layer that is intervening between the trophoblastic layer, that is the ectodermal layer, outermost layer, and the embryoblastic layer. So this intraembryonic mesoderm only further differentiates into another two layers again, which is going to become the ectoderm, mesoderm, and the endoderm. So intraembryonic mesoderm enters and it becomes one of the layers seen around the embryo. So the extraembryonic mesoderm becomes the outer coverage of the fetus that is going to contribute to the formation of the placenta, the umbilical cord, and the fetal membranes. So what are the derivatives of the ectoderm, endoderm, and the mesoderm? Ectoderm becomes the neural crest cells, melanocytes, ganglions, cranial nerve cells are all derived from the ectoderm, dorsal root, major part of the autonomic nervous system, Schwann and the glial cells, adrenal medulla, bone, its connective tissue, or that mainly the skin, nervous system, neural crest. Other than the neural crest, the other neural crest cell derivatives and the other nervous system structures, sensory epithelium, sensors for the eye, nostrils, oral cavity, all the gustatory vision and the special senses all are developing from the ectoderm. Skin mainly develops from the ectoderm. Mammary gland, melanocytes, nerve roots are all developing from the ectoderm. Coming to the mesoderm. Mainly the mesoderm is concerned with the muscle formation, bone and connective tissue formation, um, urinary, reproductive, circulatory, and dermis also, that is epidermis is a derivative of the ectoderm. Below the epidermis, the second layer is the dermis. The dermis is a derivative of the mesoderm. Now, endoderm is an innermost layer that is going to develop into the organ systems, like the organs of the gastrointestinal tract, pharynx, rectum, liver, pancreas, the respiratory system, middle ear, epithelium, the glands, thyroid, parathyroid, pancreas, liver, all of them are developed from the endoderm. So this is the image that shows you the three-layered stage, three trilaminar germ disc, ectoderm, mesoderm, and the endoderm. Embryonic period, the organ formation occurs and the placenta develops with the umbilical cord and fetal membranes. During this period, the vascular connection between the uterus and the mother and embryo is established by the placenta. So during the embryonic period, till the germinal period, we only saw the three-layer differentiation. Now, once embryonic period is reached, we will see the development of the placenta. So, the placenta is a layer of cells that are going to be helpful in communication between the mother and the fetus. So, within the placenta, there is another sac called as the amnion. The amnion is a thin membrane which envelops the embryo and it forms the amniotic sac filled with fluid. The fluid that is present within the sac is called amniotic fluid. So this is the exact image that shows you the outer chorionic villi, the presence of the yolk sac here, and see the outer cell mass has developed into a placenta, and the inner cell mass developed into the embryoblast that has given rise to the embryo. And this is the cavity in which the embryo is located, which is bluish stained color, which is called as the amniotic cavity filled with amniotic fluid. And outer to amniotic fluid, what you get will be the chorion. The chorion only becomes the inner layer to the placenta and later will develop into the placenta. The chorion only becomes the placenta later, helping it in communication with the mother. Coming to the amniotic fluid. Amniotic fluid is a very clear watery fluid which protects and acts as a protective cushion for the embryo. The embryo is suspended by the umbilical cord and laterally swims in the fluid. The embryo swims in the fluid of amniotic fluid, and the purpose of this fluid is to maintain pressure for symmetrical growth of the embryo, as well as it is also a medium for transmission. It's like a cushion to the embryo. The average volume of this amniotic fluid is 1000 ml in normally, but when it is increased, when more of amniotic fluid is seen, it is called hydramnios. And when less amount, that is less than 1000 ml, when it is absorbed, it is called oligaminos. What is amniocentesis? Amniocentesis is the aspiration of this amniotic fluid from amnion for investigative purposes. So during the development itself, if the doctor suspects of some congenital anomaly, so when they want to go in for an interventional investigative procedure to check the congenital anomaly, so they can withdraw the amniotic fluid externally and they can utilize the fluid for investigation. So the process by which an amniotic fluid is getting aspirated is called as the amniocentesis, and this is an interventional method of testing amniotic fluid for congenital anomalies. So in this what and all we can see sex-linked diseases, enzyme estimations, and we can also see the developmental anomalies, chromosomal disorders. There is also chemical testing that can be done with the markers, and it is helpful in diagnosing congenital fetal malformations. So amniocentesis is the image that shows you how amniocentesis is done. This is with the help of the ultrasonography. When the fetus is in the mother's womb itself, with the help of the ultrasonography, they identify the position of the fetus and they insert a needle into the amnion directly to withdraw the amniotic fluid. This is a method of prenatal diagnosis to test the congenital anomaly. Now let's go into chorion. Chorion is a highly specialized membrane and it is participating in the formation of the placenta. It is helpful in the formation of the placenta. It's the outermost membrane and originates from the trophoblast. Numerous small finger-like small projections that extend from the chorion is called as the chorionic villi. This chorionic villi only will communicate with the external maternal environment which is in the uh, endometrium, and this villus will form a space through which the maternal blood actually will communicate with the fetus, and the gaseous exchange, ionic exchange takes place through these villus and the intervillus space. Thereby the chorion only develops into a full developed placenta. So entire chorionic surface is covered by villi. Villi are nothing but small finger-like projections that intervene between the fetus and the mother, so which penetrate the endometrium, and they are called as villi. So these chorionic villi will penetrate the endometrium. What is chorionic villi sampling? So catheters are inserted into the vagina. So this is the image that shows a catheter getting inserted into the vagina, into the uterine cavity, where a biopsy can be taken. The chorionic biopsy is being taken, and from there the biopsy will they will remove the tissue and they can utilize it for investigative purposes to study the abnormalities within the chorionic villi, or the anomalies within the placenta. So they will also study the congenital anomalies, and fetal cells can be withdrawn through this. So through the chorionic villi sampling, even the fetal tissues can be removed. And uh, earlier in pregnancy, it is actually it is avoided. But when it is needed for the investigative purposes, this invasive method is utilized for doing the prenatal diagnosis. So, what is placenta? The structure through which mother and fetus comes into direct contact is called placenta. Is by the structure called placenta. So, it contains the bulk of chorionic villi. It is disc shaped, which is 15 to 12 centimeters in its diameter, 3 centimeters in thickness, and it is 500 grams in its weight. So, the placenta has two surfaces. One, the surface that faces the fetus is called fetal surface. The placenta, the surface which is facing the mother, that is the uterine cavity, is called as the maternal surface. So the fetal surface is usually smooth and it is covered with the fetal membranes, while the maternal surface is thrown into a number of cotyledons and it is usually not smooth. So the peripheral margins are continuous with the fetal membranes, and they are the passage through which the fetal blood vessels or the blood from the fetus communicates with the maternal blood for the oxygen exchange and the gaseous exchange. Though there is gaseous and oxygen and the ionic exchange takes place, there is no blood to blood contact there. It is only ionic exchange. So the placenta becomes a barrier of separation between the mother and the fetus. And there is no blood to blood contact. It is only the ionic exchange between how respiration takes place, how the oxygen moves into lungs. The same mechanism is applied here also. Oxygen enters, carbon dioxide is left, ionic movements takes place, but the nutrients do move inside, but there is no blood to blood contact again. So this placenta becomes a protective barrier between the mother and the fetus. So coming to the structure, the placenta consists of a chorionic plate on the fetal side, and it's called as a basal plate on the maternal side. So the plate that is seen on the fetal side, which is smooth, is called as a chorionic plate, and the plate which is seen on the maternal side, which is thrown into cotyledons, is called as the maternal side, or otherwise called basal plate. And these two plates are connected by a stem-like villi, what we call it as a chorionic villi. The chorionic villi are of two types, primary villi and secondary villi. Again, the space between these villi are called as the intervillous space, and the intervillous space is a place where the maternal blood comes into contact for the oxygen exchange and removal of carbon dioxide, as well as for the transmission of the nutrient products into the fetus. And that is a place where the exact exchange takes place. So a tertiary chorionic villus space contains fetal blood cells surrounded by the extraembryonic mesoderm. So what is a placental barrier? The placental barrier is again an important terminology which you need to which will be very frequently asked, either a 2-mark question or can a viva, or it's very important. Like, what is placental barrier? Carefully note the placenta, what I'm talking about, which is a structure where the maternal blood is transmitting its food, nutrients, and the oxygen molecules. It is composed of 4 important layers. The villus has 4 important layers. The first layer is called as a syncytotrophoblast. Then the second layer is called cytotrophoblast. The third layer is connective tissue of the villus, and the fourth is the endothelium of the fetal capillaries. These are the 4 layers present on the fetal side. So here, from the fetal side to the maternal side, there are number of layers of cells that allow only ionic exchange to happen. There is no passage for ionic exchange. Other than ionic exchange, there is no passage for the other pathogenic structures. For example, a foreign particle or a pathogenic bacteria may not be able to enter into the placenta that easily because of the presence of this barrier. The only immunoglobulin that is allowed inside the placenta is IgG. So IgA and IgG are the two immunoglobulins that are allowed into this placental barrier, and they play a very important role in the immunity of the fetus. So other than that, no other pathognomonic bacterial nucleases or pathognomonic bacterial antigens are allowed inside the placenta. So it is by nature a protective environment for the fetus. So after 20th week, the syncytotrophoblastic cells disappear and they form the placental membrane, and they are 3 layered. the layer of the placental membranes along with these four layers of the villi will constitute the placental barrier, and thereby it prevents the direct contact of the maternal blood with the fetal blood. So they become a passage through which the ions from the maternal blood are getting exchanged with the fetus. So this is the structure where this is the outer wall of the uterus. So this is the placenta. These are the villus, and you find the reddish colored spaces, intervillus spaces. This is the place where the ionic exchange takes place between the uterine blood vessels and the chorionic plate, the basal plate and the chorionic plate. So the exchange takes place within the intervillus and intervillus space, and the nutrients are getting transmitted into to the umbilical cord, and through the umbilical cord, whatever nutrient that has been received from the mother enters the fetus through the umbilical cord. It is not only the nutrient but also the oxygen molecules are getting transmitted, because the fetus does not respire and the lungs are not functional when the fetus is in the womb. So exchange functions of placenta: exchange of gaseous and metabolic products between the maternal and fetal blood in the intervillus space. Placenta prevents the entry of the pathogenic organisms from the other from mother to the fetus. So even the bacteria from present in the mother's blood cannot enter that easily into the placental covering. But there are viruses and bacteria that can pass through the barrier also. Example is the current situation. COVID, they say it is getting transmitted through the placental barrier, which needs research findings are needed. So transfer of maternal antibodies to fetus. Maternal antibodies is IgG mainly. Maternal antibody from the mother can pass to the fetus, which is protection for the fetus. The only antibody that passes is IgG. So the secretion of hormones is mainly the progesterone that maintains the integrity, estrogen that causes enlargement of the uterus and the breast. HCG is the presence of the hormone in the uterus, which is applied as an indicator for pregnancy. So human HCG means human chorionic gonadotropin is the hormone, which is the indicator of pregnancy, as well as it's an indicator for the development of the chorionic development. So HCG is supposed to be the indicator for pregnancy as well. So these are the three major hormones that will dominate the uh, uterine environment during the development. So this is the beautiful image of a fetus in the mother's womb, and see the umbilical cord. What is the content of the umbilical cord? The umbilical cord has two umbilical veins and an artery, and those three structures are embedded in a beautiful jelly-like matter, what we call as a mucoid jelly. And this jelly will be a protective covering for these vessels, and that is a passage through which the blood enters. The fetal blood with the oxygen molecule received from the mother enters into the fetus, as well as the carbon dioxide from the fetus is transmitted out through the it is exited out from the fetus to the placenta for gaseous exchange. So it's a passage for the movement of the blood. So it extends. What is umbilical cord? It is a cord that extends from the center of the anterior abdominal wall of the fetus to the placenta. From the anterior abdominal wall of the fetus to the placenta, it is going to go. It's a small cord. It is at a full-term pregnancy, the length of it may be 50 centimeters, and the cord contains umbilical arteries which carry deoxygenated blood, and umbilical vein which carries oxygenated blood. Underline, umbilical artery is carrying deoxygenated blood, umbilical vein carries oxygenated blood, just as the pulmonary circulation. Pulmonary circulation, you will find pulmonary vein bringing in the oxygenated blood, isn't it? Similarly, pulmonary artery taking in deoxygenated blood. Same way here, deoxygenated blood is taken out by the umbilical arteries, and umbilical vein is bringing in oxygenated blood. So these vessels are surrounded by a mucoid gel called as the Watten's jelly. Mucoid gel is a type of connective tissue which is actually protective for the umbilical vessels. So these are the important terminologies that you will encounter when you see your textbook. What is abortion, ectopic pregnancy, teratology. Abortion is fetus getting terminated, early termination of the fetus. Ectopic pregnancy is abnormal sites of implantation. So abnormal site other than the the isthmus area. If it is getting if it is getting on the instead of posterior wall of the body of the uterus, if the implantation is getting uh, it's happening in any other location other than that, then we call it as ectopic pregnancy. So teratology. Teratology is 4 different features of abnormality together. Ultrasonography is the investigation useful for seeing, for studying the fetal development and its abnormalities. In vitro fertilization is the lab method of fertilizing the fetus. Instead of womb, the fertilization is happening in the lab artificially. So we have also, these are the list of congenital anomalies which you will encounter in your textbook. So what is the assignment? I would like you to go through every and the congenital anomaly that is there in the textbook. Write the assignment on the congenital anomalies that are mentioned in the slide. Explain every congenital anomaly. Okay? And I will also share um, a questionnaire, okay, a questionnaire for your revision, wherein you should be able to answer the questionnaire after going through the lecture class, and after answering the questionnaire only you will give your attendance. Is it clear? Okay. Thank you.